Okay, ma'am. Warm good afternoon, everyone. We are the team here to present about COVID-19 mechanism. The team members are myself, Smita G, Narmata A, Supriya, Kavya, Nityashri, Gautam, Anantanarayanan, Arun Kumar, Shantaini, Narmata T. We are second BSc Biochem students from NGP institution. I'm gonna say about a short introduction about these are the contents. I'm gonna say about a short intro about a COVID-19. Coronavirus belongs to a coronaviridae family and a nidovirale order, and it has single-stranded RNA as a nucleic material, and it has crown-like spikes, and the size range up to 65 to 125 nanometer in diameter, and 26 to 32 kbps in length, and it represents crown-like spikes on the outer surface of virus, so it is called as a coronavirus. And past mortality were 2.9%, but present is 3.7% across 109 countries. WHO declared that the COVID-19 is a pandemic because it is occurring over a wide geographical area. And as per today's census, there are 6.73 million cases were registered, and recovered cases were 43.2 million, and deaths were 1.54 million cases. And common infection is caused by respiratory droplets, and source of infection is mainly pneumonia patients. Pneumonia affected patients will get quickly access to the COVID-19. I'll hand over it to my teammate Narmata A to continue.
Okay, Smita, thank you. And good afternoon, everyone. Good afternoon, ma'am. And I'm here to discuss about comparative analysis. Next slide, Smita. In the year 2003, the Chinese population was infected with a virus causing severe acute respiratory syndrome. And this virus is confirmed as a member of beta coronavirus subgroup, and it is named as SARS-CoV. And the infected persons will have pneumonia-like symptoms, and which leads to acute respiratory distress syndrome. A decade later, in 2012, a couple of Saudi Arabia nations were diagnosed to be infected with another coronavirus. And the detected virus was confirmed as a member of coronavirus and named as Middle East Respiratory Syndrome coronavirus. The SARS was emerged in China, and it rapidly spread around the world, and more than 8,000 people were infected. More than 8,000 people were infected, and 776 deaths were occurred during that period. And MERS-CoV was infected more than 2,028 individuals, and it caused deaths of about 838. And even this virus belongs to a beta coronavirus subgroup, and infection started from Middle East respiratory subgroup and leads to the severe acute respiratory diseases. And this is similar to SARS-CoV. Even the MERS-CoV suffers from pneumonia and renal failures. Recently, by the end of 2019, WHO was informed by the Chinese government about several cases of pneumonia with unfamiliar etiologies. The outbreak was initiated from the Yuan seafood market in Yuan city of China and rapidly infected more than 50 people. Live animals were frequently sold at Yuan seafood market in city of China. So in the year 2019, more than 50 people were infected due to this virus. On 12th January 2020, the National Health Commission of China released further details about the epidemic. This was infected as a novel coronavirus, subsequently reported in more than 100 countries all over the world. And it affects it affects human to human by direct contact and exposed through coughing, sneezing, and also through respiratory droplets. And the aerosols can be penetrated the human body via inhalation through the nose or mouth. And I hand over the session to Supriya to continue.
Now we'll discuss about the hosts of coronavirus. Next slide, please. Okay, the source of origination and transmission are important to be determined in order to develop preventive strategies to contain the infection. In April 2003, there was an outbreak of SARS-CoV-1 in Asia, that is the severe acute respiratory syndrome. The researchers found the raccoon dogs and the palm civets as key reservoir of this infection, but only the civets had a positive results of viral infection. So they are considered as the secondary host of this infection. Next is the MERS, that is the Middle East Respiratory Syndrome coronavirus. It was first emerged in 2012 in Saudi Arabia. The researchers found the camels as the primary host of this virus. Next. Next is the novel coronavirus, that is the COVID-19, which is detected in China in 2019. It is closely related to SARS-CoV-1 virus. The bats are considered to be the hosts of this virus, but actual hosts are not still known. Next, over to Kavya.
Okay, now we will see a mechanism. Sweta, next slide, please. Okay, as we all know that the coronavirus entry in the hostel is a major target for immune surveillance. Here we have done one drawing. Sweta, next slide. Actually, if you see that, if you can zoom it, you can zoom it. Once the respiratory droplet, which contains virus from an infected person, is transmitted, the main hack of human organ is alveoli. Once it enters the alveoli, its first target is pneumocyte type 1 and pneumocyte type 2. Here the pneumocyte type 1 is for exchange of gases and pneumocyte type 2 is for circulatory system, right? So once the spike proteins binds with the specific receptor, they release some RNA. You can see that side by that green color diagram. Actually, which release RNA through translation process, it produces a proteinaceous compound. That proteinaceous compound is the virus compound. So when these virus compound enters the bloodstream, they cause dialysis. They collapse the alveoli type 1 and type 2 pneumocytes. Then they increase the capillary permeability. Due to this, fluid in plasma leaked into an interstitial space. Finally, it enters the alveoli. Then it decreases the surface tension and it decreases the gas exchange and finally leads to difficulty in breathing. This is the first main cause. And the second cause is it increases the body temperature by causing cough, fever, and so on. That takes place in the hypothalamus. Then the third cause is it affects the circulatory system. First, it decreases the blood volume and it causes low blood pressure that leads to organ failure like kidney and liver. This is the overall mechanism, how it enters and how it affects their causes. Sweta, next slide. Okay. If you see the first diagrammatic representation is an virus entry where host cell protease may activate the coronavirus spike. Here, mainly the entry mechanism depends on cellular protease. Why means that virus surface has an anchored spike proteins, right? These cellular protease splits the spike proteins to mediate the entry of virus. To enter the host cell, the virus first binds to the cell surface receptor for viral attachment. Then they fuses the lysosomal membrane. If you see the second diagrammatic representation, it is the 3D structure of coronavirus spikes. There are some terms like S1, that RBD is an receptor binding domain, and S1 is an receptor binding subunit, and S2 is an membrane fusion subunit. TM is a transmembrane anchor, and IC is an intercellular tail. And I said that then now we come to a cell entry mechanism of SARS-CoV-19. Actually, how it takes place means they contain a receptor binding domain, no, which recognize an angiotensin converting enzyme to make us its receptor. This RBD, once it switch up to standing position and also comes to lying down position for immunization and receptor binding. So, moreover, to fuse membrane, these SARS-CoV spikes needs to be proteolytically activated. So, what happens? The S1 receptor binding subunit dissociates and they undergoes dramatic structural changes. Then only it enters to our alveoli. These were the features of SARS-CoV entry, which caused to rapid spread and severe symptoms and high infected patients. Next, I hand over to Nityashri.
Good afternoon. Next, we see about condition and symptoms of COVID-19. The first two common questions from the person: will the virus spread from one to another? Yes, of course, it spread from human to human and medical infection. It is very important sign. Next, second question is: are healthcare workers at risk from new coronavirus? Yes, because they are great contact with the patients than the general public workers, and it is more vulnerable to new corona infections. Next slide, Sweta. Next, we see about symptoms of COVID-19. The most common symptoms are tiredness, dry cough, and fever. And next, less common symptoms: sore throat, aches and pains, diarrhea, headache, loss of taste and smells, a rash on skin, or discoloration of fingers or toes. Next slide. Next, a serious condition, the third stage: difficulty in breathing or shortness of breath, chest pain or pressure, loss of speech or movement. Next, I will give you continued by my teammate Gautam.
Okay, good afternoon to everyone. I think you all are well aware of this SARS and it associated with coronavirus. See the slide. Okay. So let me see the genomic variations of the SARS-CoV-2. The genome of the SARS-CoV-2 has been reported over 80% identical to the previous human coronavirus, which is reported in earlier 2003. So we can differentiate SARS coronavirus 2 from SARS-CoV, which is emergence in 2002. And according to the evolutionary tree, SARS-CoV-2 is comparatively close to the SARS-CoV. And recent studies have indicated notable variations in SARS-CoV and SARS-CoV-2, such as absence of 8A proteins and fluctuations in the number of amino acids in 8B and 3C proteins and SARS-CoV-2. And it also reported that spike glycoproteins of the coronavirus, which is reported in 2019, is modified via homologous recombination. And the spike glycoproteins of SARS-CoV-2 is the mixture of bad SARS coronavirus and not known beta coronavirus. Next slide, Sita. And it also uses the same cell receptor mechanisms for the entry to the host cell, which is previously used by the SARS-CoV. And a single mutation in the SARS coronavirus 2 spike proteins may have significantly enhanced its binding affinity for the cell receptors. And the structural proteins are encoded by 4 structural genes that includes a spike, envelope, membrane, and nucleocapsids. And these genomes are made up of open reading frames, which represents the sequence of codons and that encodes for amino acids. Next slide, Sita. See, this is the cycle of SARS coronavirus in host cells. And virus begins its life cycle when a spike protein binds to the cellular receptor. After receptor binding, the conformation change in the spike protein facilities and viral envelope fusion with the cell membrane through the endosomal pathway. Then SARS-CoV releases RNA into the host cell, and then genome RNA is translated into viral replicase polyproteins, that PP1A and 1AB, which are then cleaved into small products by a viral proteinase. Then the polymerase products are a series of subgenomic mRNAs by discontinuous transcription and finally translated into a relevant viral proteins. And then viral proteins and genome RNA are subsequently assembled into virions in the endoplasmic reticulum and Golgi, and then transported via vesicles and released out of the cell. Thank you, guys. And next will be continued by our friend Anand.
Now we'll see about major obstacle in research progress. Next slide, Shweta. As we all know, in research that animal models play a vital role to uncover the mechanism of viral pathogenicity from the entrance to the transmission and designing therapeutic strategies. Previously, to examine the replication of SARS-CoV, various animal models were used, which showed the symptoms of severe infections. In contrast to SARS-CoV, MERS-CoV pathogenesis was observed in small animals. Actually, mice are not vulnerable to infections by MERS coronavirus due to the non-compatibility of DPP4 receptors. As the entire genome of 2019 novel coronavirus is more than 80% similar to the previous human SARS-like bat COVID. Previously used animal models for SARS-CoV can be utilized to study the infectious pathogenicity of SARS-CoV-2. Next slide, Shweta. The human ACE2 cell receptor is recognized by both SARS and novel coronavirus. Conclusively, Talon or CRISPR mediated genetically modified hamsters or other small animals can be utilized for the study of pathogenicity of novel coronavirus. SARS-CoV has been reported to replicate and cause severe disease in rats of a special type, that is F344, where the sequence analysis revealed a mutation at spike glycoprotein. Thus, it could be another suitable option to develop spike glycoprotein targeting therapeutics against novel coronavirus. Recently, mice models and clinical isolates were used to develop any therapeutic strategy against SARS-CoV-2, included COVID-19. In a similar study, artificial intelligence production was used to investigate the inhibitory role of the drug against SARS-CoV-2. SARS-CoV-2 infected patients were also used to conduct randomized clinical trials. Now, it is now important that the scientists worldwide collaborate the design a suitable model and investigate the vivo mechanisms associated with the pathogenesis of SARS-CoV-2. Thank you. Next will be continued by Arun Kumar.
Good afternoon. Next, now seen about the potential therapeutic strategic about the COVID-19. Next slide. No specific antivirus or vaccines have been developed from the control of SARS-CoV virus infections till day. One drug invented by the lifespan in the 2 to 4 years. Now coronavirus was a pandemic situation, so urgently the many states was approved by the co-vaccines and COVID shield also the approved at the England states also. Next is remedies were chloroquine, favipiravir, antiviral, retinovirus, and a few more repurposing for the treatment of COVID-19. Remdesivir is suitable for older people in the coronavirus. Chloroquine is a malarial medicines. India exported the 42 countries. Lipoprotein and retinoprotein is a HIV antiviral drugs. It repurposing the coronavirus. Next is Shanghai doctors isolate the blood plasma from the clinical recover patients of the COVID-19. It is a old traditional way. The plasma collected from the healthy donor of the virus, the specific neutralization with the antibodies that can be accelerate the virus clearness. So in that way, comparatively to not good. So comparatively not good. Next is antiviral agents SARS-CoV and MERS-CoV could also the repurposing the maximized therapeutic strategies against the COVID-19. Although it may have many more days from the coronavirus drug to be discovered, we must adhere to safety precautions. Next, continued by Santaini.
Now let's see about defense guidelines. Next slide. We all know that we know well that we need to wash our hands properly with running water, and maintaining good hygiene. It is also very important in order to prevent ourselves from virus attack. So whenever we are using hand sanitizer, we should make sure that that is a alcohol-free hand sanitizer, and we should cover our nose and mouth with the tissue whenever we sneeze or cough, and we need to maintain a proper social distancing in the crowded areas. Next, please. So, if you're going to the treatment or for any diagnosis, we need to wear mask, and in the crowded area, we are asked to wear mask if it's necessary. Next will be continued by Narpata.
Thank you, Shantagni. Next, a very warm good afternoon to one and all present here. Here, myself, T. Narmada from Ooty, Tamil Nadu, and studying in Dr. NJP Institute, Coimbatore, Tamil Nadu, and I like to say few vaccines for COVID-19, that is coronavirus. There is no available vaccine against coronavirus, right? Well, previous vaccine or strategy used to develop a vaccine against SARS-CoV can be effective. Recombinant protein from the Arabian strain, that is, it is a italic word of it is a strain of SARS-CoV was administered to mice and hamsters, resulted in the production of neutralizing antibodies and protection against SARS-CoV, and the DNA fragment inactivated whole virus and live vector strains of SARS-CoV significantly reduce viral infection in various animal models. So different other strains of SARS-CoV were also used to produce inactivated or live vector vaccine, which effectively reduce the viral load in animals. So there is some strain. There are some strains, TO2 and FRK, FRA. So these are the some strains. Uh, you want to see in very detailedly? I'll give a reference. I'll give the link. Next slide, please. Next slide. So, however, there are a few vaccine in pipeline against SARS-CoV-2, right? The mRNA-based vaccine prepared by the U.S. National Institute of Allergy and Infection Disease against SARS-CoV-2 is under phase of 1 trial. So, 4,800 DNA-based vaccine will be soonly available soon for human testing. So, Chinese Center for Disease Controlling and Prevention, that is CDC, working on the development of inactivated virus vaccine. Soon, mRNA-based vaccine sample prepared by Termina Therapeutic will be available. And BravoVax is working to develop a modified vaccine, Ankara, that is the MBA based vaccine. So, Clover Biopharmaceutical is developing a recombinant 2019 N COVID protein subunit termed based vaccine, and also next slide, see. Next slide. Also, research team all over the world are working to investigate a key factor, pathogenic or a treatment option. It is a demanded necessary. It is demanded necessary to focus on a competitive therapeutic option and cross resistant of other vaccines. So now let's come to the conclusion. For instance, there is a possibility that vaccine for vaccination for the other disease such as rubella and miliska can create a cross resistance for COVID-19. So this statement of cross resistance is based on an observation that children were found in children in China were found less vulnerable to infection and compared to the elder population. While children are being a large vaccinated for in China in COVID-19, which has no approach vaccine or treatment. It is a very important to prevent a spreading in the society. So prevention measurement have been discussed in a way to reduce transmission. Many studies and research are going to have a complete understanding about the coronavirus and to eradicate the virus. So, on behalf of the team, I like to say thank you for giving this wonderful opportunity for presenting our presentation and idea based on COVID-19 mechanism. Thank you. If you have any suggestion or question, you can share it.
Hello. Hello, can you hear?
Yeah.
My question is, which organ affected by the COVID-19 mainly?
Mainly kidney and liver.
Kidney and liver.
Because when it affects the circulatory system, once it decreases the blood volume, so it major causes like circulate is the heart, liver, and kidney.
I think respiratory system mainly affected by the COVID-19 because coronavirus entered through the nasal cavity to the lungs and respiratory system. So I think respiratory system mainly affected by the COVID-19. After that, kidney may be affected.
Yeah, first it affects its main hack is our lungs alveoli only. Then hypothalamus, it causes fever. Then only it affects the circulatory systems.
Yeah. Okay. Thank you.
Okay.
Any other questions, students? Okay, so I have the common comments. As you know, on the earlier presentation, I have provided that you should mention the content details, and then the reference citation should be there, and the last slides on the referencing, detailed referencing, that should be there. And I'm still trying to contact the speaker for your research methodology because it is necessary. You have to do review literature later on the given grassroots practices. So I'm trying, but as you know, due to the pandemic situation, everybody is busy and they have their own lectures to do. So